Michael McTavish up, Dr. Michael McTavish up for our next presentation. So this is the start of our science update session. So Dr. Michael McTavish is a postdoctoral research fellow working with the Smith Forest Health Lab at the University of Toronto. His ongoing research examines the implications of ecological novelty and invasion for restoration, the ecology of a non-native earthworms, and biological control of weeds, including introduced Phragmites and garlic mustard. He has worked on the first biocontrol program for introduced Phragmites since it began in 2019 in Southern Ontario. So we're all looking forward to learning more about that, and I'll let Michael take it away. Thanks so much.
Great, thank you. Good morning. Is everything coming through all right here?
Yep, looks great.
Perfect. Okay. Well, very happy to be joining everyone this morning. I want to also apologize. I'm coming off the back of a cold here, so if there's some suboptimal audio sort of situations, I apologize for that. So I'm going to be presenting on behalf of myself and some colleagues here, providing sort of an overview of the first several years of our biological control program for introduced Phragmites here in Ontario that Rebecca's teed up nicely for us here with that great introductory talk there. And so if you've joined us before, some of this introductory material is going to be familiar, and then we're going to move on to sort of what we've been doing since then. So again, Rebecca's already done a really great job of setting up some of the challenges and options that we have for managing the introduced subspecies of Phragmites australis. And we've seen situations where that can work really well, and then other situations where, for various reasons, those management efforts that we have can be limited in their efficacy, affordability, or feasibility for various reasons. And so there's always this ongoing critical need for additional options to supplement the tools that we already have from a management perspective. And so what we've been working on with colleagues for a number of years now to bring what we like to think of again as this another option in the toolbox is biological control or biocontrol. And this refers to the use of live organisms to suppress and introduce pests. And there are many reasons that biocontrol is desirable. As was mentioned, one of the reasons that species like Phragmites can get out of control is that they've been separated from some of those natural enemies that would normally help keep their population in check. So biocontrol is the process of reuniting some of those natural enemies. Great care is taken to ensure that only species that are highly host specific to that target are used, and this makes it very specific and therefore has very low off-target environmental impact. Biocontrol ideally becomes self-sustaining as the organisms reproduce, and they have the ability to move and to track the host through space and through time. And for those reasons, it can be highly cost-effective even at very large scales, again, in space and in time. So the tool that we're proposing, or that we're in the process of already adding to that toolbox then is biocontrol of introduced Phragmites. This uses two European moths that have stem-boring larvae shown here, so Archanara neurica and Lenisa geminipuncta shown as caterpillars and then as the adult moths. There are differences between the two species, but by and large, we're going to treat them as sort of the same thing for the purposes of this overview mostly. The larvae are the life stage that cause the actual damage to the weed. The larvae will mine shoots of Phragmites, and this internal damage can cause ultimately stem wilt above that feeding damage and stem mortality in younger stems in particular. Ultimately, this all comes together to reduce the competitive growth of the plant and reduce its reproductive output, allowing other vegetation in the community, ideally, to reassert itself. So to give you an idea of how these organisms work, here's a general life cycle of both of them, which again are relatively similar. The larvae hatch in the spring and will feed in the shoots for a period of about 4 to 6 weeks. And again, that's when they're doing the actual damage to the plants. They will pupate within the stems themselves after having switched through several stems over the course of the early growing season. They then emerge as short-living adults, will fly and then mate. And then the females will lay between 100 and 150 eggs or so beneath the close-fitting leaf sheaths of the plant up against the stem. So those eggs are then protected, and they'll stay in that protected environment until they hatch in the following spring. Biocontrol takes time. So this is a program that has been in development for over 20 years now, with the initial search for appropriate insects beginning in 1998. In the ensuing years, these biocontrol agents were selected, and a great deal of time and care went into host range testing to ensure that they were specific to introduced Phragmites and therefore safe to use. This culminated in a petition to release the insects in Canada and the US in 2018. The US petition remains under evaluation. It's just a longer process, but this was approved by CFIA in Canada in 2019. And so that opened the door to the current Ontario release program that we've been working on since then. So I want to provide a bit of a retrospective then on those first years of the projects and give an idea of where we've got to in that time. Broadly, we're working within Southern Ontario regionally for this. As of this winter, we've managed to release approximately 17,600 biocontrol insects. So this would be across both of the species, across 13 sites. So you see we've been able to effectively ramp up what we've been able to do here year to year. You can see those sites laid out on a map of Southern Ontario here. So you see we have some nice spatial gradients to our sites, so we're able to ask and answer various questions ecologically related to that. Because this is the first time doing this, this needs to be supported by various research objectives to develop and test these fundamental core methods to figure out how do we actually rear, release, and monitor the biocontrol agents. And I'm going to step through some key insights from each of these three points in the process. So we start with rearing. Obviously, you need the insects available to actually do anything with them. Both of the species here, neurica and geminipuncta, can be reared in the lab on either cut Phragmites stems that are harvested from the field and brought in, or on an artificial insect diet in the case of neurica. So this is a bit of a species-specific difference. Generally, we're starting at the top there with eggs, and we can allow those to hatch out into larvae that we feed in one of these different methods. Those will develop ultimately into pupae, emerge into moths, and then we collect the eggs from that. And I should say at any point in this process, we're effectively able to intervene and pull out whatever life stage we're looking at, and then choose to release at that stage. Of course, there are effects of how this rearing process works, how the eggs that are produced are overwintered and stored that will ultimately affect the survivorship of the insects, how reproductively successful they are, the timing of when they hatch, and so on. And so we have various experiments looking at all these different questions to try and improve this overall rearing and storage methodology. Once you have the insects in hand, you of course then need to actually release them onto the environment, into the environment. Both these species can be released at all different life stages. So that would include eggs, larvae, pupae, and moths. And broadly, these different life stages require different release methods to get them out into the environment. And generally, when we're comparing these, there will be trade-offs involved in the efficacy of these methods and the efficiency or the practicality. So as a very quick example for, we could look at eggs, which are relatively easy to handle. You can put out very large quantities of insects. You don't have to invest very much into developing them, but they can be fairly vulnerable, and you don't really know how they're going to fare overall. As a comparison to that, you could put a lot of effort into rearing all the way through to the moth life stage. You've helped them get to that point. You know exactly how many adults you're putting out. But of course, that requires a lot more effort and involves some of its own challenges as well. Two of the main challenges that we have then sort of generally encountered with releases that we're in the process of dealing with have to do with phenology and protection. So when we're putting out insects, these young larvae have basically preferences for the types of stems. They're looking for stems that are, for example, about 30 to 60 centimeter tall and have sort of a tissue thickness that they can actually chew into. So here we see a plot of Phragmites stem heights across different sites over that early growing season. And so you can see that if we were to go in and release insects too early, there's going to be a shortage of suitably sized stems. If we release too late, we have more stems, which is great, but they tend to be larger and a little bit too tough for them to get into. So really, you're trying to hit this fairly narrow phenological window when the stems are just right. So having ways to control that release timing is really important. The second thing that we discovered fairly on is that if you put out very exposed material, these can suffer high losses, largely due to predation. So you can see here a photo. This was just a piece of card that we glued eggs to and attach to stems in one of our very first releases. And we discovered that if we didn't do anything else, a lot of these would be gobbled up by presumably ants, spiders, and other organisms. So offering protection is important as well. So we have two of our leading release methods that we've been developing and using that address some of these questions and challenges. The first are these larval stem inoculations, where we essentially allow eggs to hatch out into larvae. We inoculate them into hollowed out Phragmites stems that we've harvested from the field, and we put blocks of these larvae-filled stems into these foam blocks. These go out into the environment. The larvae will feed, protected, on those early stems for a while, and then eventually chew their way out and move into other stems in the field. This is a lot of work, but it's worked really well as well. It gives us some control over the timing, it gives them enough food to get started, and a protected environment for those young sensitive larvae. Another method we've been working on are these egg caps. So this involves direct release of the eggs. We put them into these containers that have a screened open bottom that will keep predators out, but allow emerging larvae to climb out and make their way out into the patch. So this is much easier overall, and it does provide some protection compared to those larvae, but generally we see a lot more mortality of the larvae compared to the stem method. So again, dealing with trade-offs of different methods. And finally, once you have insects on the environment, you need to monitor how they're doing and what impacts they are having. This is important to assess long-term insect establishment and impact, and also to assess how different protocols and methods fare in the field. Because the larvae are internally feeding, and because the moths are sort of drab brown nocturnal moths, generally we're not seeing the insects directly. So typically we're looking for indirect evidence of insect damage. So we're looking for things like boreholes where they've chewed their way in or out of the stem, the wilt that results from that feeding damage, some of the secondary branching that you can see when the main stem has been damaged. To compensate, the plant will put out these secondary branches. And if we're very lucky, we'll see more directly these pupal emergence windows where the adults have made their escape into the environment. Broadly, we found that there are two really good times to look for damage. In the summer is a good time to look for feeding damage because it's fairly obvious, and then we can go back in the fall to look at more of the stem properties. The insects have sort of done their thing at that point, and we're seeing the result of that feeding. So in the summer, if it's a site where we have just released that year, we will know exactly where those larvae are going to be, so we can do a very focused monitoring centered on that release point, and we can quantify within that point and in the surrounding environment how many damaged stems we have, the types of damage, and so on. And so we can see across our 13 sites here that we have, this is kind of the initial damage that we see around sites where we have released eggs or larvae, and so we can quantify that. Typically, we see that damage on average for a site will range from about 2 to 39% of the stems immediately around that release point having damage. And this is a really nice metric because we can get it the very first year that we put material out, and we can use that to compare how our releases are going and predict how they will perform longer term. If we're looking at a site where the release has occurred 1 year previous or longer, it's a little bit more challenging because moths have had a chance to disperse, so we need to now find them in the environment. To do that, we've developed a rapid interval monitoring protocol where we essentially look at these 4 meter by 1 meter continuous sort of transects, and we're able to fairly rapidly assess whether damage is present or absent in each interval and see how many damaged stems there are per unit length of that transect. So again, if we look at those 13 sites, of the 8 sites that had releases at least a year previously, we're able to see some of this damage in at least 6 of them, which is really great. So you can see here on the vertical axis, this is the percent of those intervals that have at least 1 damaged stems. And we see that can range from 6 to full up to 100%. So seeing any amount of damage here when we go back is excellent because we're seeing evidence of overwintering survival and reproduction. And then we have a couple of superstar sites as well, where that coverage is really quite high and extends beyond the initial release, which means that we're seeing growth and spread, which is ultimately what we do want, of course. We can also visualize spatial patterns within those monitoring intervals and see how that damage tends to be clustered in those environments as well. And then finally, we can go back in the fall, as I said, to monitor more of the impacts and the consequences for Phragmites stand characteristics. I don't want to spend too much time on this because this is more useful for tracking long-term responses of populations to biocontrol. but we could do things, for example, like compare undamaged stems and stems that have been fed on by biocontrol insects and see the things that we're hoping to see, like reductions in stem height or decreases in the proportion of stems that are actually able to form reproductive structures as well. Again, not going to spend too much time on this. We're going to hear more about the responses of Phragmites and other vegetation to biocontrol. As Rebecca has mentioned, we've been very privileged to be working with Claire, Sean, and Rebecca from the University of Waterloo, collaborating a couple of sites with this Phragmites biocontrol. So we're going to be hearing from Claire on this shortly, so do stay tuned for that. So to just wrap up here, so from that 2019 start of this Ontario release program, we now have in hand these functional protocols to release and monitor the agents. We have over 17,000 insects released across 13 sites, and we're seeing very encouraging overwintering evidence and initial damage 1 year post-release, which is really what we want to see. So going forward, of course, we're going to be continuing to monitor all of these sites long-term to look for population establishment, spread, and impact. We're going to continue to experiment and iterate on the methods that we've been developing. And ultimately, our focus is increasingly going to be on scaling up. Now that we have these core methods in hand, we can really look at ramping up our rearing and release of these insects across Southern Ontario, redistributing from these nurse sites that we've been able to establish. And this is a quick side tangent that's come off some of the work that we've done here, but I did want to flag it. Do stay tuned. This is something that we're working on and hoping to have published soon. As a part of our work on the biocontrol, we've been looking at comparisons of native and introduced Phragmites, and I believe we'll be hearing more about this later today as well. We've been looking at this in Southern Ontario, and we've been putting together a sort of simple, easy to use tool that uses and identifies the best morphological traits for differentiating between these native and introduced subspecies. So this is going to be available soon. The focus has really been on making this practical and easy to use. And so we're hoping that this will be a useful complement to some of the genetic identification tools that are currently available. So that should be available soon, hopefully. Of course, this has been a large and long-standing project, so many people and organizations have been involved and have made that possible with old partners and new partners. So we'd like to, of course, extend a very big thank you to everyone who's been involved in that to make it possible. And that's it. So thank you very much. I think we've got some time for questions. We're also stay tuned for Claire's presentation, and you can get in touch with me here as well.
Great. Thank you very much, Michael. That was excellent. It's exciting to see how the biocontrol program is going, and it looks pretty successful so far. So awesome. We do have a few questions. We may not be able to get to them all, but again, I can put them in the chat afterwards. And if you're available, Michael, you can answer them there. So someone is asking, how is your program working at Oshawa Second Marsh?
So I don't want to get into too much into the weeds sort of for specific sites. A lot of these assessments sort of require multiple years to really comment meaningfully on how that population is setting up. What I will say is, so we've done a number of insect and egg releases in that region, and we have seen damage associated immediately post-release and in the time following that. So we're sort of going to keep our eye on that, and hopefully we'll have good news to report on that in future years.
Excellent. And then one last question. What is the damage to stolons, which is the primary way we see the Phragmites spread, not by seed?
Right. So, of course, Phragmites uses multiple reproductive methods to sort of spread itself and remain so competitively dominant in these vegetation communities, but stolons, of course, do play a very important role in that. The larval feeding that we see from these biocontrol moths is all above ground. So we see those damage, the stem holes and sort of the boring out of that tissue is the damage that we see above ground. However, by killing off those stems or stunting them, you are limiting the ability of that plant to acquire photosynthetic resources, which are then shunted down into that below ground environment. So there's a very tight coupling, obviously, of that above ground biomass and below ground biomass. So we're directly targeting the above ground biomass with eventual downstream impacts on below ground. So gradual suppression of those stems is sort of expected to reduce overall plant resources long term.
Awesome. Well, that's great to hear. Good news. All right. Well, thank you, Michael. Really appreciate that.